Hi everyone, I'm Irfan Tahir. I am a PhD candidate and New Harvest Fellow at the University of Vermont in the US. It's such a pleasure being here amongst all of you and enjoying all of the talks that have been happening in the last 2 days. So today I want to talk about some alginate-based scaffolds and how we were able to make them food safe using the chemistries that we do in our lab. It's right here. Amazing. So a lot of us have heard the word alginate in this conference already, and I'm really happy to see that about 4 or 5 of the groups that I've seen have been using alginate because alginate is a champion of tissue engineering. But why is alginate a champion for tissue engineering? Well, alginate is a low-cost polymer which is primarily derived from brown seaweed, and it is generally regarded as safe. So here you can see an example of the ingredient lists of a popular plant-based alternative called Beyond Meat. And you can see that the casing that is around the sausages is made out of alginate. I can guarantee that every single person in this room has eaten alginate at least once in their life. It is used in many food products, of course, not just meat, but it is also used in not just plant-based meats, but also conventional meat. It is used as a binder for conventional meat. And it provides control over mechanical properties when it comes to tissue engineering. So tissue engineers have been using alginate for decades now for various applications. And of course, because a lot of our scientific capabilities in tissue engineering are borrowed from in cultivated meat, sorry, are borrowed from medical applications of tissue engineering, we also use alginate. It also can undergo a variety of cross-linking methods. So the one that we have seen so far are ionic cross-linking, which can be done with a salt such as calcium chloride, or it can also be done with photo cross-linking, which is a non-reversible covalent cross-linking, and also physical cross-linking using a Schiff-based reaction. While alginate is an amazing material to work with, it has one big limitation, and that is that it inherently lacks a cell adhesion motif. And for those of you who might be wondering what a cell adhesion motif is basically hooks that the cells need to attach to the scaffold itself. Animal-based materials such as gelatin have inherently these cell adhesion motifs, which is why gelatin is a very popular material for scaffolds, or is added as a sort of an ingredient, but alginate by itself lacks cell adhesion motifs. So let's go to the next slide. The way I want to structure this talk is that when we started working on this, we created a proof of concept study, and let's call that the first gen of scaffolds. And primarily, I want to focus on the second gen in this talk and show some of that data. And then the third gen is the ongoing work and the things that we are doing in the upcoming months. So let's talk about the first gen. I work in a lab that is an engineered biomaterials lab, and we primarily used to work with biomaterials for medical applications. And in the medical world, food safety is not really something that they care about, nor is edibility. So given the capabilities of our lab, I wanted to create a proof of concept muscle tissue scaffold, and also no one in our lab had ever worked with muscle cells or muscle tissue before. And the way we did it, for a little bit of background on our first gen, is that we took brown seaweed in the form of sodium alginate, and we cross-linked it using calcium chloride and also visible green light, so photo cross-linking and ionic cross-linking, and we combined them together to have dual cross-linking. So here you can see the alginate structure, and you can see that to enable cell adhesion, we added a very popular peptide known as RGD to the alginate backbone using carbon chemistry. And then we also had network formation using methacrylic anhydride so that we can enable photo cross-linking. Here you can see an image of our hydrogel. We were able to create, again, like porous hydrogel scaffolds. These scaffolds were freeze-dried as well as to the previous question. And one of the biggest takeaways from this experiment was that compliant scaffolds gave us a better cell growth than the non-compliant scaffolds. And of course, these were conjugated with RGD, so we did see cell adhesion on them as well. So moving on from this, we saw some problems with this approach, which I am sure are obvious to many of you in the audience as well. One of the problems is that we are using RGD, which is a short peptide, and the food safety of RGD has not been assessed yet. So no one knows for sure if it is food safe, but we also don't know if it's if it's not food safe. And the second thing is that we are using some toxic chemicals such as methacrylic anhydride in our process. So we want to replace them with food safe and edible components. The first thing that we wanted to focus on was replacing RGD with something that perhaps is food safe. And for that, we did a literature review and we chose whey protein. For those of you who don't know, whey protein is basically a byproduct of the cheese making process. So when you make cheese, you add an enzyme to it, the cheese coagulates, and everything that's left behind is whey protein. And of course, as we know, that people who go to the gym like to eat whey protein for muscle growth. So putting whey protein together with muscle was kind of like an intuitive thing, and it is already used in a lot of our food products. So scientific literature in tissue engineering with other kinds of tissue such as bone and cartilage has shown that whey protein can provide good cell adhesion for scaffolds that are made out of plant-based materials such as alginate. So this is a detailed look at the scaffold fabrication process. Now this fabrication process is quite similar to our first gen scaffolds, but the difference is that we have replaced RGD with whey protein. So here you can see that we have our whey protein isolate, which is a highly concentrated form of whey protein. We attach methacrylic anhydride to it. The NMR plot on the top right confirms that our conjugation of the methacrylic anhydride was successful. And then this undergoes photo cross-linking using green light at 525 nanometers. And here we have basically three material groups. We have the methacrylic alginate without any whey protein isolate. Then we have the non-modified version of whey protein isolate with methacrylic anhydride and alginate, so ALGMA. And then we have modified versions of both whey protein isolate and ALGMA. So we have the three material groups here. One of the first things we did is that we did physical characterization. And here we looked at the swell ratio and the weight loss of our scaffolds over a period of 24 hours. And here, one of the key takeaways from this slide is that we saw that the non-modified version of whey protein isolate actually did not stay inside the scaffold after 24 hours. And you can see that the weight loss for the WPI/ALGMA group is the highest out of the three. And this sort of gave us an indication that one advantage of modifying the whey protein isolate with methacrylic anhydride is that it's able to stay inside the scaffold in a better way. And this was confirmed with our ISCCM images where we did see porous scaffolds, but more important than that, the scaffolds were denser for the one where we modified the whey protein isolate. Another advantage that we saw of modifying the whey protein isolate with MA is that we were able to create scaffolds with lower stiffnesses at the same concentration of alginate. So here you can see that actually the blue one is the modified whey protein isolate and also the modified alginate. And this gave us a scaffold that was physiologically relevant to a muscle tissue, as we saw in the previous talk, that the stiffness of muscle tissue is about 12 to 15 kilopascals. So again, this kind of chemistry enabled us to reach those compliant scaffolds. And following the trends of our first-gen scaffold, we saw that more compliant scaffolds gave us better cell growth. So we saw a similar trend even in this project, in this second gen as well. And you can see that for this, the more compliant a scaffold is, the better the cells are able to grow on the gels themselves. So these are C2C12 mouse muscle cells. And I'm happy to, of course, say that for our third gen, we have graduated finally to bovine muscle cells. And it is far more relevant to the cultivated meat industry. And of course, the cell metabolic activities also show that the modified version of whey protein isolate were actually not toxic to the cells themselves. So let me just bring you back to the three generations of scaffolds. With the first generation of scaffold, this was the first time in our lab that we were working with muscle cells. We wanted to use the chemistries that were available to us and create a proof of concept muscle tissue scaffold. For our second gen, we replaced RGD with a food-safe cell adhesion motif. So we verified the food-safe cell adhesion motif. But really, I would say that the bulk of our work and perhaps the most exciting results of my PhD are coming in the third generation of scaffolds. When I started my PhD, a lot of people told me that really you're going to do the best work at the end of your PhD. And of course, looking down, I didn't really believe them. But now I'm starting to see that because I have the skills, because I've already spent a lot of time learning all of the techniques, I'm starting to get really, really good results. So with the few minutes that I have left, I want to give you a little bit of a sort of a preview of the things that are ongoing in our lab right now. So one of the things that we tried is that we did directional freezing, which is a very popular technique in tissue engineering where you're able to create scaffolds with grooves inside it. And actually, as Diana said, and I've never thought about it this way, one of our previous speakers, we're creating these irrigation channels inside the scaffold so that the muscle cells are able to grow inside them. And this is done in a very simple way using a liquid nitrogen and a steel plate. And as the steel plate freezes, the scaffold is able to freeze in a certain direction. You can see an image of our third gen scaffold, which is entirely food safe. It has a whey protein isolate in it as well, and a comparison of that with decellularized skeletal muscle tissue of porcine. Another thing is that we are improving the ways in which we look at cell adhesion. So you can see that we are now able to look at the cells on gels at a closer microscope level, so using SCM. And then using bovine, we can see that from day 1 to day 3, there is a really high cell expansion. So again, that is, of course, also a positive result for us. And this video, which shouldn't be elongated, actually is a video of me changing the focus on the microscope and showing that the cells were actually able to go inside the scaffolds and migrate inside the scaffold itself. The scaffold is about 2 millimeters. It's a bulk hydrogel scaffold. So the cells don't just attach on the surface of the scaffold, but they actually go inside the scaffold itself. So overall, really, those are some of the highlights of the work that we've been up to. So in the first gen, we just wanted to create like a proof of concept, but with our second gen, we improved one major thing about the scaffold. But for this, the third gen, we are aiming to create an entirely food-safe, edible scaffold that we can seed bovine muscle cells on and then able to grow that in a directional way so that when we differentiate those cells and the cells go from being a myocyte to a myotubes, then there are mechanical cues that we can provide to the cells themselves. At the end, I would like to acknowledge our small team at the University of Vermont. So my advisor, Dr. Rachel Floriani, the PhD student, Chris Foley, and then our lab manager, Patrick Sharon. I would also like to thank all of the funding organizations, the University of Vermont Graduate College, New Harvest for funding this project, and also my entire PhD, and Millipore Sigma for also giving us some funding for lab equipment. And thank you so much.
Thank you. Are there any questions? I was already waiting for that.
I
Just
don't
go
want
ahead.
to be the
Yeah.
weird one, but okay.
No, it's okay.
So thanks for the great talk. Did you compare the whey protein samples with RGD samples as well? Because in the pictures, I just saw that your control was like pure alginate, but I don't know if you had as a positive control, like with RGD, to see how would that compare?
Actually, I'm doing that like right now. Yeah.
Okay.
But RGD is something that, yeah, I mean, do you think RGD would be a good positive control?
I mean, at least standard control,
Think
because
so?
you want to replace RGD, right? And then look at like the pictures. I mean, okay, it is positive compared to not having whey protein, but is it like enough to completely replace RGD? Probably you could also look into whey protein to see if it has the RGD sequence. Maybe it has. And then that could already help to better understand the system.
That is definitely something we're planning to do because there are papers, because we chose whey protein isolate, but there are many other cell adhesion motifs that people are looking at. For example, pumpkin seeds, chickpea, and that is one technique that they do to look at the protein concentration and to see if there's a match with some of those RGD components.
There is one patent on replacing RGD with a plant-based that they already actually did. It's also from dairy food industry. But I don't know if whey also has that sequence or not.
Be worth looking at. Thank you so much for the suggestion.
Catch of the day. So one issue with whey protein might be that it's still animal derived, which might not go down well with everyone. So is that something you consider? And shouldn't the long-term strategy be get RGD approved for food safe applications
I
instead?
was really hoping that you would ask that question. I came
My
prepared.
pleasure.
So whey protein by composition is the highest protein in whey protein is beta-lactoglobulin, which we can now produce using precision fermentation without animals. And we can produce it at mass. In fact, in the US, you can find it in grocery stores, in ice creams, cream cheese, milk, protein powders. So there are some companies who are doing this, and we plan on collaborating with them and using precision fermented beta-lactoglobulin rather than whey protein isolate. However, I would like to preface this by saying that I am from the dairy state of Vermont in the US. And the fact that we are using a byproduct of the cheese making process is something that the farmers and the agriculture industry there is very excited about. So we do have some collaborations with the farmers there. So I would say we should use both.
And cost-wise, making the whole protein versus only a short peptide sequence, which could do the same trick. What's the outlook there?
I think we should work really hard on making RGD food safe as well, because RGD is something that the chemistry of that is well defined. There are a lot of labs in the world who use it. So if RGD has sort of like the label of being food safe, that would be a win for the cultured meat industry. However, that is not the case right now. Right now, whey protein is food safe, and we have all, most of us have eaten that.
Thanks.
Yep.
Thank you very much again.